It's a pleasure for me to tell you today some of the work that we've been doing. Those who know me from the campus know that I've been very interested for many, many years to interface with the nervous system. And the idea is, how do we design systems to either record from the nervous system or to stimulate the nervous system? And over the years, I've come up with various ideas how to do this. And so today I will talk about some of the latest work that we've obtained in the somatic nervous system, which has been a long endeavor in the lab, but also I'll tell you some of the latest work, which is more recent, on how to interface with very small nerves of the autonomic nervous system, which actually poses a very specific challenge because these nerves are very, very small in diameter, very fragile, and difficult to record from. So I'll start with some of the work that we've done in the somatic nervous system, and this has been a long lasting project. In fact, I'm trying right now to renew an R1 the 5th time. So this has been going on for 20 years plus. Same project. And you can see the idea right there. That is, it's called the flattening nerve electrode. And started that a long time ago in the lab. And you can see that I can use the laser, I guess. The idea is to take advantage of the fact that, and actually we had to fight a long time for that concept, that nerves are not round, even though everybody thinks that they are, and they can be round, but they don't have to stay round. In fact, a lot of nerves in the body tend to take the oblong shape on their own if you squeeze them between your fingers, or if you dissect them off the muscle fascia, they would naturally go round, but if you flatten them up, they readily go into this shape. So the idea, of course, is that if you have a flattened shape, then the circumference to cross-sectional area is a lot larger, since a round shape is the optimum minimized circumference for a given surface area. And therefore, you can put more contacts. And you can see an example of a shape of electrode that we use for selective stimulation of this fascicle or that fascicle. This is the same concept, but adapted for recording. For recording, what is needed is a much longer structure that has maybe 1.2, maybe 2 centimeters long. And it has various shapes. You can see in the middle, these contacts here are placed exactly in the middle between 2 reference electrodes. These reference electrodes are usually connected, and the signal is measured between this point differentially using these 2 as the reference. And there's a third reference somewhere else. This design has been working quite well, and I'll show you some results that we've obtained with this idea. And the purpose of the length is to account for the fact that when action potentials come down through the nerve that's sitting right there, they tend to go quite fast, and so the wavelength is quite long. And in other words, as we measure the voltage difference between this point and this point, by the time the action potential is gone, the voltage can be small. So you need to have fairly long structure. And this design, this differential design, is done in order to remove the EMG. The idea is this point and this point are connected. They are at the same voltage. And therefore, any EMG difference between this point and this point that is along the nerve is minimized. Not zero, but it's minimized. And since then, we developed new methods to remove EMG. And EMG, as you can guess, is a huge problem because the signals from the muscles are in a several millivolt range, whereas the signals from the neurons are more like 10, 20 microvolts. So that's the basic idea of this design, and it's been quite successful. Over the years, especially from some people in the audience, we've been able to, you can see an example of a nerve that's actually quite flat. This is a femoral nerve in a human. And you can see a large number of fascicles. And it's possible to place an electrode around there. And here's one, I think, from Dustin Tyler group, where you can actually selectively activate groups of fascicle one at a time. So he's presented some of this work before, so I'm not going to spend time on that. And he has gotten some really interesting results using this design to restore sensation in patients, and I'm sure you're all familiar with this concept, so I won't spend any more time on that. But you can see that these are all fine electrodes placed on 3 different nerves using the same concept, although these electrodes are not as long as the one I showed you because they're not designed for recording, they're designed for stimulating. And that simplifies the situation a little bit. But I want to focus on more of the recording aspect of this problem because it is quite more complicated than stimulation. The idea there is if I have a cuff electrode, and this is a plot, this is a simulation, okay? If I have several fascicles, in this case 2 fascicles, one here and one there, and if I place electrodes up and down 2 sides after I flattened it, can I recover the origin of the signals? Where are the sources? Not only that, but it's a good thing to know where they are, but it's more important to know what they're doing, what they're saying. So this is a problem that's been on my mind for many, many years. And here's some of the work that has been done by Brian Woodlinger. And you can see that the voltage recorded along this side of the electrode and on the other side of the electrode is plotted. So the top one is plotted here, the bottom one is plotted there. You can see there are quite a bit differences in voltages here and there. It's not too surprising I have a source here and a source there, right? So maybe there is some kind of an algorithm that we could use to recover the source, the location, and the intensity of the sources. So this show you an example of how this can be done. Here's the actual exact position of the sources, and this is the center of mass of the electrical activity recovered at this point. This is what's known as inverse problem. How do you recover signals on this, inside this, from known voltages on the outside? So this is sort of encouraging, and then we ask the question, yes, it's one thing we can recover the signals, but we can recover the location of the source, but are the signals accurate? And here's an example of how we test this idea. This is also done in simulation. This was published in J&E 2011. Here's an example of a signal simulated. This is a simulated electroneurogram. You might be surprised to see that the signal looks very stochastic. And it turns out it is stochastic. The statistical properties of ENGs have been studied by Sasho Jasonek, who was actually here at Case Western quite a few years ago. And he said, basically, you can take a single unit recorded outside from a single action potential. You can simulate that. Fairly easy, a few microvolt, 10 microvolt, 20 microvolt action potential. Then you add a whole slew of them with a random delay. And that is basically an ENG. And you can see that when you reproduce that signal right here, it actually looks somewhat like an ENG. So we did that. We actually placed that signal in the fascicle 1, and then another signal in fascicle 2. They're independent stochastically, right? But they're overlapping, which is important because we want to know if it might be easy to record or recover a signal when there's none on the other fascicle next to it. But when they're together firing at the same time, maybe it's very difficult to distinguish them, right? And so we, with this simulation, confirmed a point that we've seen by several of the students, And the idea is this. If I look at the separation between two fascicles, and now I look at the cross-correlation between the known signal and the recovered signal by the algorithm I told you about, that cross-correlation increases as the distance between the dipoles, or the fascicles increases. It makes some sense. If they're separated in space, it seems like they would be easier to recover. And we end up with actually quite interesting results, because you can see the 0.95 cross-correlation is excellent. And you can see that's about 1.5 millimeters. You can stand at 0.9, at about 1.2, etc. So as you go down to 1.8, the signals are much more difficult to recover. But this simulation shows that there is some hope, and this is actually happening while these two signals are overlapping. So how does it work in real life? This is an example of a recovery. This is also Brian's work. This time it's in rabbits. Can't remember what we picked animal model, but I think it's because it's a little bit easier to deal with than cats. We were actually quite surprised with these animals as we tried to do the stimulation of the ankle. We couldn't find more than 1 degree of freedom in an ankle joint of a rabbit. And I was telling students, something's wrong with your electrode. You must be missing something. And then when we talked to the vet said, no, there's only 1 degree of freedom in the rabbit's ankle. Well, only cats and felines have this complicated ankle joint with 3 degrees of freedom. But you can see that in this case, where you have the peroneal and the tibial branch and the main branch here, it's possible to recover the signals, and the signals recovery is with the electrode is placed. These are 3 different rabbits. The electrodes are placed on top and the bottom, and this is in color the recovery from the peroneal here and the tibial here in red. And superimposed is the anatomy of the nerve, and you can see that these two match quite well on all 3 cases, even on this one where these 2 fascicles are actually quite similar, very close by in location. So the location recovery is actually working, but this is an acute animal, right? So the animal is sitting quietly. There's no EMG. All there is maybe some music in the surgery room, and that is it. It's very quiet. So then the challenge is, can we do this in a freely moving animal during long periods of time? And this project was actually funded also not just by NIH but DARPA. Jack Judy at the time was very interested was a director of a program there, very interested, and noticed that there was basically no data long term. People do experiments for maybe a week, 2 weeks, 3 weeks, but we need a device that actually works for very long times, months, years. So he was pushing to have experiments that will last a long time, and we took the challenge and we accepted the money and did the project. And the idea is this. Here's an animal. This is subject. These are mongrel dogs. And I picked these large dogs, because they have ankle joints with fairly large nerves and simple nerves, maybe through 3 fascicles, that's it, simple. And the challenge is, can we recover these 2 fascicles as the animal is walking around freely on the treadmill? And here's the design of the electrode. It's very similar to what you saw. The only difference is it was adapted in collaboration with Dustin to include a much thinner silicone sheet, so it's extremely flexible in the longitudinal direction. So in other words, it can bend fairly well, but it's stiff in this direction so that it maintains its flat shape. And the way we do this is by adding on the electrode this yellow, that's a polymer that's a little bit stiff, just a little thickness of polymer. And you recognize the reference electrodes on both sides. These are the middle electrodes. etc. So this electrode is about 2 centimeters long, as I said. The height is only about 2 millimeters or 3 millimeters tops. So it's a fairly long structure, but it's designed to be quite flexible. Now here's some of the 3 prototypes. I showed them a progression of this at different times, 2012, 13. One change we made here is a strain relief for the wires as they come out of the electrode. We found a way to make sure, because the first time we did this, they were breaking right there, and we found a way to loop them through so that they wouldn't break. And here, the last version, the last addition to that, this design worked pretty well originally, except for this relief. That's one problem we had. But we added in yellow a foil, metal foil, gold. And this, the idea is on the outside of the electrode, and it's exposed to the external solution. The idea is that it forces this plane on the outside to be grounded at 0, and it minimizes the effect of EMG, and it worked quite well. This idea was tested by one of my previous students. You might remember Paul Yu, who is now at the University of Toronto. In fact, just got tenure last week, I think. And he showed that on the computer this should work. So we tried it, and we have some very good results with that. So we since then implemented it. So here's the electrode in question during surgery, implanted on one animal. You can barely see it actually. And we've submitted that electrode to tests on the computer, this finite element modeling, and also we tested it for its ability to expand a little bit. If the nerve wanted to swell, we need to accommodate that. So we did some experiment with measuring pressure inside as the extension, whether as the pressure inside is increased. We developed our own percutaneous plugs out of titanium based on some of the ideas we've seen in the literature. And they worked somewhat, some Netix connector in the middle. And this is designed. One problem is that we did not really want to go all the way and place this electrode on the head of the animal because that's a little too far. So we placed those somewhere in the back, somewhere here. So they are still moving around a little bit on the skin. And I still think it's a problem, but it sort of worked. The skin would clearly cling to the titanium, which is right here. And if we monitor the animals regularly, we were able to maintain a certain degree of quality of the signals. Here's some of the Cheney staining showed very little effect of the presence of this electrode on the nerve, but I think I have some more data on this. So one of the big issues that we had, and that's bugged me for a long time before I even started this, is the electronics. You might think, no, this is not a big issue because there are commercially available electronic amplifiers that can do the job. But the answer is no. And the reason I knew that is because 15 years ago, there was another student, Masoud Sahin, working in my lab. And I'm sure Tom remembers that very clearly. And we were recording from the hypoglossal nerve, which is a nerve that passes in your neck here and controls the movement of the tongue, to detect possible episodes of apnea, obstruction. And at that time, we designed a really cool, I thought, very low noise amplifier based on the fact that if you match the source resistance of the electrode to the power of to the ratio of the voltage to the current of the referred noise of the amplifier, you can really minimize the noise. You need a transformer. It's complicated. It's external, but it worked. And what we saw as signals that were really small, clear signals, but very small, sometimes less than a microvolt. Now that is really tiny. And so I knew this would be a problem because that dog they were beagle dogs working with hypoglossal. They were in a Faraday cage up in the dental school at the time, and they were not moving, but I figured that we'd have a lot of problems with the dogs moving around to detect very, very low signals. So we searched, and you can see here it's hard to find an amplifier that has very low noise and high bandwidth, because the bandwidth of the ENG signals is up in the 10 kilohertz. The larger the bandwidth, of course, the larger the noise. So we came up with an idea. I came up with this idea of it's called hardware averaging. I'm sure you're all familiar with averaging. You take a bunch of signals, you put them in, you add them up, and anything that's random disappears because it subtracts, where everything the same stays. But you could do the same thing in hardware. This is a software problem. But in hardware, if I take an existing amplifier that has maybe this is from Intent, for example, has 120 or 64 channels input. Each one of these has a noise of about 2 microvolts, which should not be sufficient to see anything in the cuff electrode. And you average them, you will decrease the noise by square root of the number of channels. All you have to do is connect them in parallel. It's very simple. And so this idea was tested, and here's built right there. We actually built an implantable version of that, but I'm not telling you about this, but the idea actually worked really well. And here's some data on these amplifiers. You can see that, here's a noise of one of these Intent channels, which is a state-of-the-art recording channel right now. They're actually designed for a neural signal. They're capacitively coupled already, and they are perfect for this type of application, but still look at the noise. It's still pretty high, 6 microvolts, and that would mask everything that you want to see. If I take 4 of them in parallel, the noise goes down. Put 8 of them, it goes down even further. We're now down to plus or minus 2 microvolt peak to peak. It's starting to get better. And here's some of the recordings that were made in the dogs. You can see how the noise decreases as we increase the number of channels. And at some point, we're down to, say, 1 microvolt RMS here, which is quite good. So it's an extremely simple way of doing it. Of course, there's a price to pay for that, because the more channels you have to average, then you have more supply. We came up with an algorithm to optimize the number of channels and try to minimize the power. Here's the power spectral density of these amplifiers. You can see how it goes down very quickly as you increase. But there's a limit, right? I mean, eventually at 16, the improvement doesn't get much better. But anyway, this idea worked, and we implemented it. So the next thing we had to do is improve the algorithm. And so we had tried several algorithms. I'm going to pass on the details, but I have several students over the years try to figure out how to recover the signals. And we never really did not have a really good way other than the computer to know whether it was working or not. So this is a work of Tom Eggers, who graduated, defended his thesis last year in December, and he will get his degree in May, I believe. He built this rig, and here's a fine electrode right there. And he placed this wire here that contains this structure that contains 2 wires. One is here, de-insulated at this point, another one here, de-insulated. So that forms a dipole. That dipole is placed inside the cuff. We can move it around anywhere we want, place a source there, and then record signals from there and try all the algorithm we want at our heart's content. We can find out if they work better, if the dipole is close to the edge, close to the source, wherever. We can put multiple ones, etc. So that system was developed, and we used it to test the algorithm. And the one that we currently working with the electrode that we have is called the hybrid Bayesian. It has a long history in some extent because the original algorithm, which is this equation, was developed by another student of mine, surprisingly, Sri Nagarajan, who is currently the head of the MEG lab at UCSF. His algorithm is called Champagne, and he uses it to recover sources from the brain. As you think of something, can you find out where that is? So his algorithm, we adapted it for this particular case, and I'll skip on the algorithm exactly, but basically here's the two dipoles in the cuff electrode. We use this algorithm to recover all the different sources. And this is the noise, and these are the cross-interfering sources. And I don't know if this is working too well. I think the battery is dead. But maybe I can use that. And so here's the two dipoles here. They're separated. We measure that exactly. Here we put this signal into one. We put that signal into the other. Again, you see we have an overlap between these. And we try to recover them. And you can see that there is pretty good correlation between, well, we don't measure it, but you can look at it between the recovered signal and the original signals. They follow each other, and we can basically recover these two. And if we quantify all that, it turns out to be quite well, working quite well. So now we ask the question, we implant this in a dog, and is it stable? Can we record over these animals, from these animals over long periods of time? As I mentioned, these are 3 mongrel dogs. And what I'm plotting here is SNR. And this is the baseline noise. And this is the contact impedance. And you can see that, sorry, it's quite stable over time. The SNR is in the range 4 to 6 or 8 sometimes, in the more like 5. So it's not very high, but it's still pretty good. You'll be surprised when you actually see the signals that we're getting that kind of SNR. The baseline noise is consistently below 1 microvolt RMS, and in some cases even down to 0.5, which is, I'd say, is some kind of record on a fully moving animal. I don't think it's ever been done. Keep in mind also that the source electrodes have their own source resistance, right? I mean, they're not infinitely big, they're small. The smaller they are, the larger the impedance of the electrodes are. That by itself creates its own noise that you can go around, because that's Johnson noise. It's just the noise of an impedance. And so these numbers include the electrode noise and the amplifier noise as well. And the impedance of these electrodes were quite stable, and this impedance I was telling you about is about 2K or so, which creates by itself maybe 0.4 microvolt RMS, 0.3, 0.4. So the noise, you can tell from this that the noise of electronics is almost 0. And also look at the duration of these, right? 6 months. We went to 8 months. Something happened on 9 months here, but very stable up to 8 months. So it's just quite remarkable. So here's some of the signals. You see, I'm only showing you one of these here, but there are 16 channels that look like this, identical, that we recorded continuously as the animal is walking. We also have here some ways to detect the position of the angle of the ankle optically. And so we measure ankle joint, and we measure ENG of these channels as they're walking around on the treadmill. We did have one issue with the treadmill, though. The treadmill, even though I was very careful picking it out, still the motors was creating a lot of noise on their own. So we had to basically turn off the motor and have somebody walk behind the animal and then move the thread. But you got to think about this, right? You got to find a solution to this problem. So then you do this and you put it together, and this is what you're seeing there on the bottom is an image of the electrical activity inside the cuff electrode as the animal is walking, reconstructed in time and in space. So you see plantarflexion, dorsiflexion alternating as the animal is walking. And you can see the person walking behind I don't know if you saw that. See, the person here is walking behind the dog to move the treadmill, because otherwise but we know how to solve the motor noise, however. I haven't implemented it, but there's a solution to that. The solution is to put the electronics right on the electrode, and so there's no induction possible. So I think this is shows you that it is possible to recover signal. These animals, there's no Faraday cage in there. The experiments are carried out in the animal facility somewhere. I don't know, somewhere here. And there was noise. There was no special thing we did to do this recovery. So it's possible. And here's an example then of the signals that were recovered. And this is dorsiflexion source, plantarflexion source shown here while the animal is walking. And at the same time, of course, we also did EMG recording. I did some cross correlation between the two signals. And I'm not sure I have any data to show you, but this is the next thing I wanted to do, and this is about to get published hopefully soon, is to get some idea of the information transfer. It's been done in brain interface machines, but it has not been done in nerve machine interface. So I wanted to know how much information is actually passing through from the nerve into the electrode and into our recording system. And this is done for a single fascicle, the tibial nerve. And what you see here is a plot of the tibial nerve versus the EMG in the gymglossus muscle. And as you plot that, you see very strong correlation, of course. It should be, if you're recovering our signals properly, you could, you expect that. Except that a while ago, one of the first student published a paper showing he could recover either dorsiflexion or plantarflexion, right? So it's a binary, so one bit, right, of information. So the next question, can we recover more than one bit, right? And if yes, how many bits can we recover? And you can see that it's easy to tell. You just put a grid and figure out, place a bunch of bits here on this plot. And we were able to recover about 8 beats per second on between all the way to 8 weeks. So 5 bits per second, 5 or 6. And that is quite similar to what a brain-machine interface would do. So and it's only for one fascicle. So, I mean, it's not going to drive very sophisticated 10 degrees of freedom artificial arms, but it shows that it's possible to quantify. There is some clear information there. This is actually proportional, right? Sort of proportional between here and there. So it's possibility of a proportional controller, not just a on or off controller for an artificial device based on the information that we recovered from these fascicles. Can we do this in humans? Well, this is going back a few years where we took the femoral nerve and asked how many channels can I recover from this multifascicle nerve, and you can see we recovered 5 easily fascicles from this. This is the computer stimulation, not a real experiment. But look at the size of the signals. I mean, they're pretty small. So now I'm going to switch to the autonomic nervous system. And the question there is, can we use that same technology in one of the part of the nervous system that has been sort of neglected for a long time, and I'm guilty of that myself. I was thinking of nerves, I'm thinking sciatic, I'm thinking femoral, etc., but I was not so much thinking about vagus, and yet the vagus nerve, which passes by your neck, innervates every part of your body, and it's mostly actually afferent fibers that come in large numbers from your gut, and the gut contains a huge number of neurons, believe it or not, that's on the surface of the gut, and it's like having another brain basically down there, and nothing is known about this, very little. What are these neurons doing? How do they communicate? They all talk through the vagus. So we have access to the vagus nerve because it's not that far away from the surface of the skin. So in principle, some of this technology should work. And so the one I described earlier. But when we were approached by a company called GSK, who was very interested in doing some of this work, and they put some very stringent limits on what they wanted to give us the money. We want you to record from a vagus of a rat. Vagus of a rat is only 200 microns in diameter, and there's no way you can put a cuff electrode to record selectively from different parts of that nerve. So I knew this would be very, very hard, and we decided to come up with a different technique. Looked at what's out there right now to record from nerves other than the cuff electrodes. And one of these techniques that you'll be familiar with is the life electrodes, the longitudinal intravesicular electrode that was pioneered by Ken Horsch, been taken up by Yoshida, and groups in Europe are using this. Basically, you take it's actually the first one was a Kevlar thread, pretty small, that was covered, metallized, and then insulated, and sewn into the nerve with a needle. Since then, they've used polyimide with multiple contacts, and the time electrode is another one that's pretty popular in Europe, where they take a very similar idea, but they push it right down to the nerve. And there are a few other electrodes, but not many really. So then I looked at this, and I knew that it didn't work well. And didn't work well because I thought they're too big or too stiff. Wasn't quite sure, but they didn't last for long, and they still don't. So you look at the literature and you find out how do I know whether this electrode mechanical property matches with that of the nerve? And everybody's talking about the Young's modulus, of course. The Young's modulus is a measure of stiffness. But we, as a group, decided that we discussed this project that stiffness is not the best thing to measure. What's needed is flexural rigidity. Flexural rigidity is a product of the second moment of inertia times the cross-section diameter of the structure. It takes into account the Young's modulus, but also the geometry of the device. Clearly, if you have a wire that's very long and it's easier to bend in certain dimension, and using the Young's modulus by itself doesn't make sense, to me anyway. So we started to look at this. Flexural rigidity actually is a well-known measure in mechanical engineering, and but nobody had used it in this context. So nobody measured flexural rigidity, and to determine if the interfaces that we actually put in nerves are similar to those of the nerve. So we measured it. Well, actually, it's not measured. This is an estimate based on the dimension and the Young's modulus. 10 to the minus 9. The time electrode, 10 to the minus 9, 99, 10 to the minus 12. This is for a platinum iridium wire, 10 micron, very thin. These wires are quite flexible, right? 10 micron. We use them a lot also, but they tend to be a bit brittle. But you can see the flexural rigidity is 100 times, so 10 to minus 10 about. Parylene C is in that range too. Silicone is very flexible. Carbon nanotubes is what we figured out that we should use. But now look at the biological flexural rigidity of various structures. One of them is the rod photoreceptors. These are tiny, and this flexural rigidity was measured using lasers, optical techniques to move things around. You know, you can, the laser tweezers, you can move things around a little bit and put some force. The right sciatic nerve is, this is a mistake, it's more like 15. So these are small compared to these. So in other words, these electrodes really are several orders of magnitude larger. So then we asked the question, can we build a system that would have the dimension of an axon and the mechanical property of an axon? Very similar. Can we just build another axon, basically, and stick it in there so that the nervous system doesn't even know it's there? That would minimize all the reaction and fibrous encapsulation possible. So you can see that here, this is the typical nerve. Recognize the axons. The fascicle here has the perineurium around it. And here's the epineurium. And so we've envisioned this wire that's really small, about 10 microns, similar to an axon, that would be finding its way through the perineurium and into the bunch of axons that are there. And in principle, if that works, we should be able to put it into any fascicle from any nerve, as many as you want. So take this nerve, which has maybe 20 fascicles. You should be able to put an electrode in each one of them, maybe 2, maybe 3 even. So this is a way of doing multiple channel nerve recording, if it works, right? Nerve recording that would have the capability of extracting signals from multiple fasciculated nerves. So that was our vision, and we worked quite hard on that the last few years, and we made a lot of progress to implement this vision. And we use, as I mentioned, the carbon nanotubes because they are extremely flexible. So let me tell you a little bit about carbon nanotubes. They are made here in Cleveland in the lab of Liming Dai, micromolecular science. And the way it's made is very interesting. There's a substrate there, metal. You coat this with the catalyst, and then you put this structure into a chamber which contains, I think, carbon, hydrogen, etc. And slowly, from the base of the metal, from the catalyst, rings of carbon will form and slowly rise up from the bottom, not from the top, from the bottom. And eventually you end up with the surface covered with these tubes all over the place in high density, about to a length of about 500 micrometers or so, not very tall. Then you take it out. I saw it done. It's very fascinating to watch. Then you pull the corner a little bit and you twist it. So these carbon nanotubes, because of van der Waals forces, will bend down. So if I pull from the corner, it will bend down, and you keep following, you twist them, and you end up with a yarn, like this. And if you look at this in more detail, this yarn is made of individual nanotubes. Each one is about 500 micrometer long about. So even though the nanotubes themselves are very stiff, and very strong, the yarn is not, because it's made of all these individual nanotubes. But it's extremely flexible, and it has a huge surface area, of course. Therefore, it has a very low impedance. And if you look at this plot here of, in red is a carbon nanotube impedance as a function of frequency, and in green is a platinum iridium wire with the same diameter, and you can see there's a factor of 1 order of magnitude lower. So we use these tubes, this yarn that we get once in a while from this lab. We insulate them with parallel C. We leave the tip exposed. And now you have this extremely flexible wire, extremely flexible. You can't even, you just barely see it. And it's moving around like crazy. How on earth do you put that into a nerve? So that was a challenge. So meanwhile, though, we did measure, that's the work of Yang Zhang, who's did their master's measuring the flexural rigidity of these tiny structures, extremely flexible. And that was a challenge. We spent a lot of time. We eventually found that we could use atomic force microscopes, which have a picoforce measurement system. You apply force on the yarn, and you measure displacement. From these measurements, you can actually extract the flexural rigidity of these tiny wires. And you find it's in the range of 10 to the minus 12 or 11. So that's quite flexible. So how do you put them in? Well, we came up with this idea. And at the time, I knew about something, a therapeutic technique called macroneurosurgery. No, neurography, sorry. And if you take a tiny needle, and a neurologist will do this, you take this tungsten, very sharp tip, about 2 micron diameter tip, and then place it under, with no visual guidance, find a nerve, put it into your arm through the skin until they get into a nerve. They leave the electrode in there, it's called macroneurography, and then they record action potential passing through units, and they check the velocity, whatever, for diagnostic purposes. This is a fairly well-known therapeutic method, or diagnostic method, I should say. I don't think it's used so much anymore, but I did talk to neurologists, a very expert at this thing, and they said, yeah, I can find the nerve. Does it cause damage? I don't think so, because if I kill axons, the patient would jump up and down. So apparently, he told me, don't use a sharp electrode, use blunt, but very small diameter, but blunt. So that's what we did. Took the same needle that the neurologists use, exactly the same, and Frederick Hare. And then we wrapped the electrode here around the tip in such a way that there is a knot at the very end there that holds it in place. Now you have the electrode, you put it into the nerve, it has this shape in there, and you take out the needle, and it's gone, right? And then the wire stays by itself. Then during surgery, we surround the whole thing with fibrin sealant to hold everything in place. And voila, as they say. Back home. So here's an example of an implant, such an implant. This is a vagus nerve of a rat. You can see it's sitting on top of 2 hook electrodes, this one and that one. And automatically, right, it takes a flat shape, right? I mentioned that earlier. Nerves are not round, they're very plastic. And normally it's much narrower than that if it's a round shape. But you can see we implanted 2 of these such electrodes, one here, one there. They're clearly shown in there. It has a little twisty, helically shaped tip. So it tends to stay in place because once you put it in there, it sort of resists movement, which is a great advantage because when you have surgery, you want to put everything's moving around. Once you put electrode in there, you don't want that thing to come out easily. So that helped a lot. So here's an example of that. And we put the fibrin around there to close it up. And here's some of the signals that we get. So this is very first recording chronic in the vagus. In fact, the first that I know of, more than 2 days. The record so far, I was told, is only 2 days, semi-chronic. So people can record the hook electrode very short periods of time. This is 15 weeks post-surgery. And look at the size of the signals. SNR is very large. And you see this burst of activity there. You blow them up and clearly units show up, We're right next to axons, so you can see individual axons passing through. Oops, sorry. This may be what's known as post-hypoxia response, because here at this point we induce hypoxia, but we don't know for sure. The reason we don't know anything about these signals is that nobody's recorded from them. Somebody in the audience one day I showed this, asked me, did you fast the animals before you did this test? I said, I don't know. Well, then they're probably from the gut. The gut is contracting, trying to do something. So these are maybe part of the enteric nervous system. We don't know. Very hard to tell. But clearly, there are units, because if you look at them in more detail, here's an example of that. They look, they walk like units. They must be units. The problem we have is interpretation of those signals. Let me show you an example of a signal obtained from the vagus in the rat as it's being anesthetized. So it's undergoing anesthesia. I'm skipping chunks of time, right? But you can see that the activity is slowly increasing, and in fact is matching the respiration. So we spend quite a bit of time trying to understand what those signals are. For a while, I thought they were connected to sinus arrhythmia, but I don't think so now. I think they're more, they are lung afferents responding to isothoric. So during anesthesia, they get excited, they fire away. Since the vagus is full of afferents, then very likely these are afferent responding to anesthesia. But we don't know for sure, and you look at the SNR, it's quite hard in this animal. Okay. Now we have also done experiments in another autonomic nerve called the carotid sinus nerve, or actually the carotid sinus nerve. This is what we wanted to do with this. Is so small that we decided not to, but here's a carotid sinus nerve here. It's about 100 micron in diameter. It only has about 400 axons in it. It's tiny. And yet, it's quite important nerve, right? Because that nerve carries information from the carotid sinus. It contains the baroreceptors, chemoreceptors, and goes right to the brain, and then controls heart rate, response, blood pressure, etc.
Is it a rat or is it in a person?
It's in a rat.
So
Oh, yeah. But see, our challenge was to work with rats. The GSK was adamant that you got to do this in rats. This work was actually funded by NIH, and we could have done this in patients, I suppose, but I doubt it. So here's some examples of the recording there. We actually recorded blood pressure at the same time. And the blood pressure was done, we got a supplement from NIH to do a blood pressure measurement system with telemetry. So we implanted a sensor directly into the femoral artery, the abdominal artery. And we're able to record for the first time carotid sinus activity and blood pressure. And you can see some of the units that we're recording. So these are actually directly from the carotid sinus nerve. Since then, we sort of abandoned that and look, we're recording from the glossopharyngeal nerve, which contains the carotid sinus fibers. Carotid sinus is a branch of the glossopharyngeal. And you can see a very strong correlation between the activity here and the blood pressure. So it suggests that this is working, and these recordings are obtained from the glossopharyngeal nerve, and we were able to look at the signals here, right? Pretty nice quality. We're able to distinguish between two classes using classification on the units, the standard method. And this is the work that was done with Shane Q. Joe Marmstein was working on this, and Grant McCollum was working on this. And you can see that there are basically two classes of units that respond to hypoxia, and we classify them and name them based on the physiological response as chemoreceptors and baroreceptors. And they seem to fit the pattern of the physiology. It's hard to be absolutely sure. This was published fairly recently in 2017. And this is, in my knowledge, the first time anybody is recorded from baroreceptors and chemoreceptors ever. We also did some test stimulation. This electrode with this high surface area, you would think might be adequate for stimulation. In fact, there's been some papers on that before. The charge capacity of that electrode is quite high. And we implanted one of these electrodes, several in the sciatic nerve. I think it was a peroneal branch. And Tom Eggers did that. And look at the charge threshold required over long periods of time. This is very stable to produce the same response over long periods of time. So I think the stimulation part is sort of the easy part of that electrode. One more difficult thing is the histology, because once you have that electrode in there, it's very small, very hard to find afterwards. But it can be done. So what we decided to do is to use this new technique, histological technique called Clarity, that was just started in the brain, coming out of the lab of Carl Dysseroth. And it hadn't never been done in the peripheral nervous system. It turns out that Joe Marmerstein, who's sitting over there, had some experience with that, and already in the lab in WashU. And we decided to apply it to this. And the advantage of this is that if you want to cut a nerve that contains a implanted device, you can do that. But in this case, you would have to cut the wire with it. And we got several people to say, no, I'm not doing that. I'm not damaging my knife. This is just carbon nanotube. You can cut through. No, I'm not doing it. So we said, okay, let's leave it in there and do the clarity, clear everything, and look at the whole thing, the whole structure with the wire using a 2-photon microscope. And it makes plenty of sense, in fact, because if you really want to observe the reaction next to the electrode, you don't want to remove the electrode, which would kill all that, because we don't expect too much of a reaction. So this is an example of a vagus nerve that was cleared. It's almost transparent. And that actually took quite a while, maybe 2 weeks to clear that. You put them in the solution. But since then, we talked to Michael Jenkins, who's here in Cleveland, and he has developed in his own lab a clearing procedure. I don't know if he's here today. A clearing procedure, oh, there he is, that is much faster, and it's working, according to us. In a couple days, we can clear it. It's a secret recipe, which is fair enough. We don't care, just want to use it. But you can see some of the tests that we've done. DAPI, you can see the electrode here. You can almost guess where the encapsulation is. It's very thin. This is 10 micron, right? So the encapsulation is only about 10 micron on each side, very, very thin. There's hardly any reaction from the Schwann cell still there, etc. So we couldn't see anything obvious in the histology. So I'm just going to conclude here that it is possible to record from multiple fasciculated nerve and recover information from these nerves using these cuff electrodes. It can be done in a freely moving animal, and signal to noise ratio, SNR, is in the 5 range or so. But we can also do this with these tiny electrodes for fascicles that may be difficult to record from, particularly if you have a fascicle in the middle of the nerve and it's surrounded by other fascicles. It might be tricky to extract information from the surface of the cuff. in that case, perhaps a combination of a cuff electrode plus a wire might be the solution. You can pick out the fascicles you want to inject with an electrode. So maybe a hybrid solution in the end would work. So as you can see, this interfacing is evolving over time, and hopefully I'll be able to continue some of that work in the future and then try to implement these hybrid ideas. Some of the people who worked on this, you know them all, so I'm not going to mention. You might not know Daniela Calvetti. She is a professor in mathematics, used to be the chair for quite a while, and she worked with us on some of the algorithm that we developed. And you may not know Brian McLaughlin, who worked at Draper, and he was on the contract with us from DARPA. And Yazan, you know him, he's now a professor in Jordan, and he's coming back, I think, next summer. Just got married, and hopefully we get to see him again, I don't know. But Grant is here, and Pedro Musini, you may not know, he's a double professor and also worked with us a little bit on that. And it's funded by all slew of places, DARPA, NIH, NSF, GSK, etc. Thank you so much for your attention.
Thank you, Dominique. We have a little reminder for you.
Well, thank you so much.
Remember your visit to Cleveland?
Yes.
Are there questions for Dr.
33 years coming. Yes.
Are there clinical applications? Are there clinical applications for recording in the human vagus nerve? Like, do people envision some sort of feedback system where recording from these systems
So
are?
right now we're working on epilepsy.
Yeah.
The vagus is heavily involved in epilepsy. In fact, there are a couple papers, acute papers, suggesting that maybe you can actually predict seizures from the vagus. We are very interested in that. And there's other papers of great interest where you can maybe measure glucose levels for patients with diabetes 1, where you can measure glucose. These are some of the examples.
Do you need the electrode that's so small and flexible in the human vagus nerve?
I think you want it as flexible as possible. Yes, why not? The question that you want to ask
Hard
is,
to make as well. They seem challenging to make and procure.
yeah, the procurement is tricky, but in principle, I don't see any reason why this could be implemented. It could be a factory. I mean, I've seen it. You just buy equipment and make it. It's not a research tool. It's an accepted technology. So I don't see that as a major problem. I mean, you could replace it with the platinum wires. We did a lot of that work. 10 micron platinum wire work also, but there's a big difference with the carbon nanotubes is that they're more brittle. We treat them exactly the same way, but they tend to break more often. So carbon nanotubes really make a big difference. They are flexible and they don't break so much. So can it be done implemented in the patient? Yeah, it's sort of like optogenetics. I mean, eventually, maybe. If this shows it can be done by a surgeon, for example, fairly rapidly, I'm picturing a multifascicular nerve, say the median nerve, and the surgeon goes in and places one of these things everywhere, like 10 fascicle, 20 fascicle, in like half an hour, connect them to a connector and done. So you can record and stimulate. This is particularly important. I didn't talk about much, but one of the big challenges is recording sensory information. It's been very tricky. In fact, the cuff electrodes, how good it was, you saw that the thing is quite good at recording signals. We got very little in terms of sensory. Some, I skipped the slide, but there's some sensory information there, and we don't really understand why, because the motor signals, the signals that we saw, we think are motor, but maybe not. Maybe they're also sensory. They just happen to be related to the motor function, right? So sensory recording is tricky. So now we're moving hopefully to a pig model. The pig has, strangely enough, has nerves that look very much like humans, like a tiny a mini pig, a Göttingen mini pig, but not that big, right? Like this big, has an ankle, a sciatic nerve with 30 fascicles, whereas a dog has 4 or 5. And I don't think the pig's ankle is so sophisticated. You wouldn't think so, right? But when you look at it, it has 4 fingers. Where the dogs, I don't think have that sophistication. Canines, yes. But, so the pig is much more sophisticated than we think, I think. So that's what we'll try to do.
Very interesting. Question, are you looking for any inflammatory?
Yes, we are, yeah. But we're
so
just
you
at the beginning of that.
Yeah.
So we're in the
But
process
that
of doing
seems to, that's the biggest problem.
Yes, of course, yeah.
Inflammation.
But we've seen so far very little of it, and the response to on the published work that we have shows very little. But it's still preliminary because we need to do it in more animals. We need to be more specific about inflammatory response, etc. But those wires are very small, and if you look at the literature in the brain in particular, you find that the idea of implanting extremely small structures makes a difference in the response. So if you implant a Michigan light probe that has a pretty wide structure with multiple contacts, compare that directly to the carbon fiber, which is very much in vogue right now, you see a big difference. And so, and I believe the difference is because these carbon fibers are maybe less than 20 micron in diameter. They're about the size of a neuron. There's no cytokines accumulating around there. They can diffuse freely through the tissue, whereas if you have a surface, perhaps, that's an hypothesis, right? Then cytokines accumulate and kill and damage tissue. So these number nanotubes are very small also, so I don't expect a big response.
So I was struck you did one of these recording nanotube electrodes one at a time, and you couldn't distinguish between the source, like maybe you did finally, but how would you use this for discovery? Like you said, nothing's known about
Yeah. No, that's
this.
We're
Yeah.
struggling with that, okay? We're struggling with that question. The way to do it is to challenge the system. So you're recording and you want to eliminate, say, all the heart, the gut, and just look at the lungs, say. So you put histamine in the lungs, you have to anesthetize the animal. So right now we're focusing on one, this is some of Joe's work, is trying to find the cardiac component of the vagus. And one way to do it is called coherence averaging. So we average the vagus just at the QRS. And we look at the response of the vagus and when the heart pumps. So it eliminates everything that's unrelated. So now we're focusing on the cardiac response. And we see a signal there. We don't know what it is again. And I asked my buddies in cardiology. They don't know either. They don't know what it is. Nobody knows. Yes, Michael.
I was just going to say for the discovery part, I don't know how to do this, but maybe if there's some way
But
to
you're going to say it anyway.
track. Yeah. But I mean, if there's what you really want to know is you want to know the axons you're recording from, where they're going to, and maybe if there's some way to combine this with track tracing, maybe if you coat the electrode with dye or something like that, that could tell you where those are going, in the vicinity of where the electrode is. Maybe that could help or something.
Yeah, you can
like
mark
that.
the axons. There are some, but one of the things that we don't even know is, and really wants to know is, are these afferents or are these efferents? That simple idea would, what you're saying would tell us about that, which would be nice, because that's an important thing. Is what we see is a response coming from the brain or is it coming from the organs? That in itself is important. We don't know. We can't distinguish purely based on the recordings.
And that's because of size.
No,
No.
it's just because there's no size difference or there's nothing different about the signals. Afferent and efferent are the same diameter axons, as far as I know, and so we can't distinguish them.
Any other questions? Thank you very much, Dominique.
All right, thank you so much.